we will be holding the second summer seminars series, Science Never Stops, and Dr. Ogün Adebali will talk about his recent research findings on the COVID epidemic. Dr. Adebali received his bachelor's degree from Middle East Technical University in 2011 and his PhD from University of Tennessee in 2015. And he worked at different research labs at Oak Ridge National Laboratory and University of North Carolina at Chapel Hill at his postdoc positions. And since 2018, he's with the Sabancı University as assistant professor. And he got international and national Young Scientist Research Awards and installation grants, and recently awarded with TÜBİTAK International Fellowship for Outstanding Researchers. Let's welcome Dr. Adebali, and it will be an exciting talk. Yes, let me give it my position to Ogün. Okay.
Thank you very much for the kind introduction. So it has been exactly 2 years since I started at Sabancı University. So I started on June 1st, 2 years ago. Thank you very much for being here. So today I'm going to talk about basically how we can use evolutionary information in our daily life, in our research life, and why we are interested in evolution and how we can use it for the recent SARS-CoV-2 issue. And I will also mention about our recent work about that. So let's get started. I would like to start with this quote from Tadeusz Dobzhansky. So he says, nothing in biology makes sense except in the light of evolution. So this is quite true to me, because if you don't understand evolution, if you don't look at from evolutionary perspective, then many things will be senseless, and that's why I really like this quote from him. Another quote is going to be from Bill Gates, and what he says is, DNA is like a computer program, but far, far more advanced than any software ever created. So actually in our lab, we try to understand how this DNA works basically, but we benefit from evolutionary information. We benefit from how evolution works, and we try to understand how DNA works by using this information. And briefly, what is bioinformatics? So I was lucky to get this definition from Wikipedia. Now it is open. An interdisciplinary field that develops methods, approaches, and software tools for understanding biological data. So what is key point here is it's an interdisciplinary field to understand biological data. This is what we need to know about bioinformatics. And we, as a computational biology lab, we benefit from bioinformatics also. So, and this is very basic, but I thought it would be nice to go over the basics in this presentation. So this is the central dogma of biology. So basically we have DNA as the genetic information, and this DNA is converted to RNA, and then RNA is converted to protein, and proteins are the fundamental units of life, of cell basically. And if you want to copy this DNA, it is possible through a process called replication. That's why we have this here, but this is a very sort of an abstract view, abstract sort of dogma of biology. So there are, of course, many complications to that, but we have to know about this. We have DNA, and at the end we have proteins, and all the information that we need for proteins are encoded in DNA. So this basically shows you the journey of human genome sequencing. Of course, it starts with Mendel, and father of genetics, I would say. Then one of the milestones is in 1953 with the Rosalind Franklin, Watson, and Crick discovering the double helical structure of DNA, and then series of experiments and sequencing. So sequencing starts with Sanger, and then we have next generation sequencing today. Human Genome Project started in 1990. Then in 2003, we have the finished version of Human Genome Sequence. It is now completed. Thanks to sequencing technologies. And after 2008, we have next generation sequencing technologies. Then we can sequence any organism with no problem at all. And eventually, we will see sequencing more and more in the clinics. So because each of us has a genome, so that it is, if you want to know about your DNA, you have to sequence your DNA. And this is the genetic material in the cell is called genome, so that once you sequence genome, you know about the DNA sequence. However, what it means is still a question, and we try to understand what this DNA means. Actually, the first genome was the bacteriophages genome. So it was way back to 1976. So this is the first genome completed. And after that bacterium, another bacterium, Haemophilus influenza, this is a bacterium, and its genome was completed. And then today we have our human genome in hand. So not only human genome, but also the sort of entire landscape. If you just look at the life, and we have organisms and genomes from every sort of domain of life, I would say. So this is the tree of life. What you see here is the three kingdom of life. One is bacteria, the other one is archaea, and the rest is basically all eukaryotes. So these are the three kingdoms of the life. So it means that we have genomes from each organism here, right? So, and the phylogenetic tree shows evolutionary relationships between these organisms, and the depth of the tree, if you look at this scale, it shows you the time scale. So millions of years, 5 millions of years to 4 billions of years. So about 3.5 billions of years ago, life started with bacterium, then it split it up, archaea evolved, then eukaryotes evolved, and today we have the current organisms. So this is called phylogenetic tree, and each organism is represented as a leaf of a tree, and we have internal nodes and branches. So you can think phylogenetic tree as a regular tree. It has a root, it has branches, and it has leaves at the end, and leaves represent what we have today, but the nodes, the branch nodes, are the common ancestors of the leaves that the branch has at the tip of each branch. So this is the evolutionary information that we can basically infer from the genome sequences. But how it can help us in our lives. So basically, I would like to talk about what we do, what we want to do in our group, and how we can use evolutionary information to understand biological processes, right? So I would like to talk about some sort of subfields, basically. So, you know, I talked about genome sequencing. So after you sequence the genome, what can you do about it, right? So first, you have two options. First, you can go to the lab and you can try to characterize the proteins and genes. And the second alternative is you can annotate that genome. How that annotation works, you just compare the genome of interest with the one that you find in the database that is already well characterized. Because it is possible that it will have a relative that is well characterized, you can basically compare these two genomes. And if you just put edges between the regions in the genome, then you can basically characterize them using the evolutionary information. So just like here, you have chromosomes from one organism and chromosome set from the other organism. If you can put edges between them, if you can show the homology relationship, and homology means that two entities share a common ancestor, then you can infer about their functions and protein functions, for example. This is one use of evolutionary information. The second use would be characterizing the microbiome. So what is microbiome? So believe me or not, we have in our body, 90% of the cells are microbes, basically. We live with bacteria. So basically, microbiomes is the communities of these bacteria. You can find microbiomes everywhere and bacteria anywhere. So if you just look at this carpet, there will be tons of bacteria. But some bacteria are not cultivable, meaning that you cannot take this bacteria and take it to the lab and let them grow because you don't know anything about that bacteria, right? So what can we do about that? So luckily, we have now a new field called metagenomics. And with metagenomics, you don't have to culture bacteria. All you do is you just isolate DNA from wherever you want, let's say the carpet, and then you sequence those DNA pieces, and then you characterize them. And how you do the characterization, you again benefit from the evolutionary information. You just look at the similar sequences, the annotated ones, try to understand what sort of organisms do live on that piece of carpet. So this is how we can use the evolutionary information. What else? We can identify protein-protein interactions. So let's say that proteins work together in the same biological pathway. Let's say that they do have interaction with each other. Then they have to be together in the organisms. So this example shows you the swimming ability of the bacteria, right? And these three proteins, green, red, and blue one, they are necessary to perform the swimming function. It is called flagellar movement and signal transduction. That's basically, it is called chemotaxis. If there's a sugar, bacteria wants to move towards sugar. If there's a repellent, like a chemical bacteria move away from that repellent. So these three proteins, if you look at the tree of life, they are either together or they are absent together, right? So their presences, co-occurrences, and co-absences do correlate. So you will either see none of them or you will see three of them together. And in evolution, there is a sort of a rule. If you need something, you use it. If you don't need don't need that thing, you are free to lose it because mutations happen all the time. And if there is no evolutionary pressure on losing a protein or gene, then you are free to lose it as a bacteria because it doesn't come as a cost in the fitness. So what we have here is the co-occurrences of these three proteins. And with these co-occurrences, we can infer protein-protein interactions. These interactions can be physical interaction or functional interaction, but at least functionally, they interact each other because they are working in the same pathway. What else we can do? We can predict protein interaction sites. How it is possible? Let's say this protein A and protein B, they do interact, but we don't know anything about their interaction site. At which site they interact, right? We don't know about the interaction interface. So how we can predict that? If these two proteins interact, their interaction site should have some sort of features that should be more or less conserved. And if you look at the amino acids, and if you look at the evolution of those amino acids, you will see that co-evolving residues or amino acids are there in protein B. So to maintain the interaction in evolution, if a substitution occurs in one protein, another substitution compensating that original substitution could have also occurred to maintain the interaction between these two proteins. So this is basically what we see. These blue regions, blue amino acids, blue positions basically, are co-evolving with the red one, the green ones, and then you can predict, by just looking at the co-evolving residues, you can predict protein-protein interaction. So, and then this is another view of showing which sites, just looking at the evolutionary information, you can accurately predict protein-protein interaction sites, and which is sort of fascinating to me. Also, protein folding is a sort of big mystery of biology. How one sequence, amino acid sequence, can fold into protein and then it starts functioning. So it's still largely unknown how it happens. There are many sort of theories about that. But can we predict that by just looking at the evolutionary history of that protein? basically the same idea, co-evolving residues. If two residues interact with each other in three-dimensional space, then this interaction should be conserved, so that if one amino acid changes, another compensatory amino acid should have changed also, and then the interaction, in 3D space, that interaction should be maintained. And this is what we see here. This position is co-evolving with this position, and because they are interacting in the three-dimensional space. So what we see here is the predicted, by just using evolutionary information, predicted 3D structure of proteins, and this is the correct, the real structure, crystal structures of the same protein, and you can actually assess how good the prediction is by just comparing these two visuals. And finally, we can use evolution information to predict disease-causing mutations. So let's say that we are interested in one amino acid substitution, and we don't know whether it is causing any disease or it is just neutral. So most of the substitutions that we have today, they are neutral. However, some of them increase the chance of having a disease. How we can predict those? Again, evolution is our friend. We can look at the other organisms, and you can go back to the common ancestors of animals, for example, and see if that specific substitution occurred anytime. And if, for example, let's say the first D, the aspartate, is human, and we have chimpanzee, primates, mammals, vertebrates, and invertebrates. So if there is no D to K substitution here, aspartate to lysine substitution, then it means that in evolution, for millions of years of evolution, this substitution did not occur. It means that if you have that substitution, you will have something wrong in your body. This is what it means. However, if a position is sort of substituted to lysine here, just like here, so you have many substitutions in the evolution of this protein. If this is the case, then you conclude that, okay, this substitution is allowed in evolution, so that you conclude that this substitution could be a neutral substitution. So why I tell about this is because this is how we use evolution in our daily life, not your daily life, but in research, these sort of fields, subfields use evolution information quite a lot, and without evolution, without evolutionary perspective, the life would be much harder for us. And now I can talk about SARS-CoV-2, why we are interested in that. Actually, I thought about showing this to you or not, but it basically represents what happens. So lab members, including me, so we have our own projects. They are funded projects, so they are nice. We are excited about that. But this SARS-CoV-2 issue emerged, then we are sort of distracted. So if you didn't know the name of this meme, it is called Distracted Boyfriend. And now we, as a lab, distracted with this SARS-CoV-2 because it is very interesting and we want to know more about it. Just like any of you. So if you are here today, probably you want to know more about SARS-CoV-2. And we as humans are very interested in the things that basically life-threatening, right? So if something is threatening us, so we want to know more about it, like our enemy. You want to know more about your enemy, and that's why many people are interested in this subject, including us. And we use evolution, we use evolutionary methods. So if this is so, this is virus and virus is evolving, we said that why not? And why not use the methods that we already use in the lab so that we can understand more about that. I'm not going into detail on what happens and when happens. All of you, I'm sure, know about the basics, but on January 7, the World Health Organization officially announced this virus as SARS-CoV-2. They named this virus as SARS-CoV-2 that causes COVID-19, and it was well known that it was a coronavirus family. We know about coronavirus because 6 of them, so now we have 7, 6 of them cause common cold, basically. SARS, MERS, they are also among them. They are not common, but you know, we remember them from 2000s. And then today we have more than 6 million cases worldwide. So remember the central dogma of biology. So normally we have DNA, and then at the end we have protein. In this virus, it is not exactly the same way. We don't have DNA in this virus. What we have is RNA, and RNA replicates itself, and there's a direct and indirect translation to protein in this virus. So this is the process of how virus works. So DNA is not there, but RNA as a genetic material is there. So this is the sort of the genome of the virus. So it has around 30,000 nucleotides in its RNA. So open reading frames, basically normally one open reading frame encodes for one protein, but this virus is sort of different. Again, one protein is encoded from single open reading frame, but there is proteolytic cleavage so that there will be 11 different proteins basically encoded by ORF1A, ORF1B, and the rest of the proteins, each of them is one open reading frame. So these are called non-structural proteins, and these are called structural and accessory proteins. Why? The ORF1AB, they basically produce proteins for replication of the virus. So they are not needed for the structure of the virus. So this is a structure. So, you know, phospholipid bilayer, actually they are taken from humans, from hosts. And then the rest is basically the proteins, spike protein, capsid protein, envelope protein, and so on. But none of these proteins are part of the structure. That's why they are called non-structural proteins. And the spike protein is the important one. Why is that? So this is the more realistic view of the SARS-CoV-2 developed, actually created by Jeremy Smith at Ogün National Laboratory. I remember taking classes from him. So you see the spike protein is sticking out. Actually, it recognizes the host receptor. In humans, it is called ACE2 protein, ACE2 receptor. And then this is the spike protein interacting with that receptor, and then virus can enter the cell through this interaction. So human ACE2 is here, and this is the spike protein. There are details, we don't need to go into them, but basically this is the key interaction between the virus and its host. So I would like to give you an example actually posted in New York Times, so it's a nice visual, to show how we can follow the mutations to identify the transmission routes. So one example here is this position. In this position, one RNA letter changed to U, right? So uracil is there because this is RNA instead of thymine. And then another substitution happened at another position. And this position also changed the protein amino acid here, right? So isoleucine is now our new amino acid. And this position, again, the substitution changed the protein structure. So some substitutions do change the protein amino acid, and some substitutions do not. So the ones that do not change the protein amino acid, we call them silent mutations. And for example, ACU, which is a codon, we call 3 letters as codons. And when this U is mutated into A, then it won't change the threonine substitution, threonine amino acid. So it was threonine, after mutation, it is still threonine. Most likely, it will not affect anything about the biology of the virus because the protein is same. However, there are some non-silent mutations. And for example, if this C is mutated into U, then this threonine will change to isoleucine, and it is called non-silent mutation. And this will change the protein. If you look at the number of amino acid substitutions found in 4,000 something coronaviruses from December to April, you will see that some positions are having many, many substitutions, and some positions like this will have no substitution. So what does it mean? It means that the positions having more mutations actually are tolerating new substitutions, right? So it really doesn't matter whether it has one amino acid or another amino acid, because probably it is a structural placeholder amino acid, so that it won't cause a drastic change. However, if you look at the spike protein, the red one is spike protein. Remember what it was? So it was important for the host receptor interaction. This site has no mutation at all. So what does it mean, right? So mutation, we know that mutation is a random process, more or less, right? However, we don't see any mutations here. How it is possible? Actually, there were mutations, right? So virus replicates, and there were mutations here. However, the ones having mutations in this region, they were not be able to survive. They were not efficient in spreading. They were not efficient in sort of transmitting to many people, because of probably the interaction site was disrupted. So that's why today, when we isolate the genomes from all around the world, we see no mutations here. The reason is this region should not change. Remember how we use the evolution when predicting the disease-causing mutations. The same logic applies to here. It means that this should be a conserved region. It should not be mutated to any amino acids. So that's why this is a conserved region, and it doesn't tolerate substitutions. Because for virus, it is disadvantageous to have other amino acids on that side. And let's say that there were these mutations, and this is the mutation set, and another mutation set. If you have one genome from Wuhan resembles what we isolate in Seattle, then we can basically predict the potential transmission from Wuhan to United States of America. So this is how we identify the relationships between the genomes, so that the transmission routes. I would like to give you more details, more examples on that. So imagine that we have A, B, C, D viruses, and each diamond represents a mutation. So all of the viruses here have this blue mutation. So it is not informative for us. So blue mutation doesn't help us to distinguish between viruses, right? However, only 3 of them have the pink mutation. Only 2 of them have the yellow mutation, and this green is specific to C, and this purple is specific to D virus. So if this is so, how we can cluster that? How we can build phylogenetic tree out of these? We can basically cluster A and B together, because we know that these 2 viruses have this same yellow mutation, and they look very similar throughout the genome, so that we can cluster together. So A and B, it is most likely in the common ancestor of A and B, if you look at the phylogenetic tree, this mutation occurred, and then A and B viruses actually separated from each other with this yellow mutation in their genome. And this, remember, C is here with A and B. Why is that? Because of this pink mutation. So pink mutation was common to ABC, and then the common ancestor of ABC here, this is where mutation happened, then it split it into AB and C lineage, and this green mutation is specific to C lineage. And in the common ancestor of all viruses, we have this blue mutation, and then this purple mutation is specific to D. This is how we can cluster the viruses into groups, and how we can basically identify the evolutionary relationships between the virus genomes. So this is phylogenetic tree. It is fine, and the x-axis normally shows the substitution rate. Now, is it possible to transform this phylogenetic tree into time tree, which means that x-axis now is the actual dates? Luckily, we can do that because we have the information about the dates of sample collection, right? So we know that C is collected later than AD, so that we can adjust the branch lengths, and we can have the time or date as an x-axis now. So we can basically transform from phylogenetic tree to time tree. And we also have location information. So location 1 is all blues, location 2 is the orange one. So if you have such a view here, C is location 2 and blue is location 1. What is the most probable scenario? So most probable scenario is that from location 1, there was a sort of transmission to location 2, because C is the only one coming from the location 2. And the ancestral version was in location 1, and then one of the patients here probably moved the virus to location 2, and that's why we have C in the location 2. And this is how you can basically identify the transmission routes. And we should remember, I should remind you that not all samples are sequenced. Only a very, very tiny subset of the genomes are sequenced. So that's why we may have missing links, but still, we can have a clear idea of the transmission route. So these patients here are not sequenced. So we can only infer about them, right? A, B, C, D are sequenced. Today, this is what we see. But we can infer about the history and about the evolution of these viruses by just looking at A, B, C, D. So for example, this yellow mutation happened in the common ancestor of A and B. And this was specific to D. It doesn't have to occur in D. It probably happened in another patient, which was the common ancestor of D. And same here, A, B, C have a common ancestor here, and this is where ping mutation happened, and so on, right? And probably not C, but another person moved from location 1 to location 2, and this is where the location-specific mutation happened, lineage-specific mutation, and today what we have C, but C doesn't have to travel from location 1 to location 2, right? Another patient, which is the transmitting one, could have most likely traveled from location 1 to location 2. And about the techniques, briefly, we basically need to align sequences, the virus sequences, RNA, right? So I would like to give you an example of what sort of mutations that we can have. You can think the RNA as a sentence. So for example, this one, I will be by the lake is the sentence. And mutations can happen. And for example, this E can mutate into W here. I will be, now D is having a W instead of E, and lake. So does it change the meaning? Maybe not. So if you look at this, you will just understand that, okay, this is probably a typo, so that you will understand the meaning maybe. But some point mutations change the meaning. So here, we see L to C mutation, I'll be by the cake. So now we have a totally different meaning. And this is a nice analogy with the sequences, because one mutation can change the function of a protein. And there are some other types of mutations like nonsense, frameshift, nonsense basically, and immature stuff of the sentence and so on. So we have many of them, and additional to them, we have insertions and deletions. So if you look at here, I'll show you an example of deletion here. I will be the lake. It means that by is deleted from this sentence. Now it is I will be the lake. Now, or I will be by the lakeside is another sentence. Now we have an insertion, side is inserted here. So how we can assess them? We have to align these sequences in a way that we can infer their homologies. So how do you do that? I'm sorry. So here, side was inserted here. That's why we have to put dashes here in the other sequences to see that insertion. And we have to put dashes here to see the deletion in this sentence, so that all of the amino acids or nucleotides will be aligned together. Because this D or B, they share common ancestor. They are derived from the common ancestor. That's why we have to align them all together. The aim is, in multiple sequence alignment, the aim is to align the homologous sequences with each other. And then we build phylogenetic tree. I'm not going into detail. Basically, we compare sequences to infer about their evolutionary relationships by predicting the common ancestral versions. There's one critical point here is the rooting. So normally in tree generation, what you have is such a tree. It is unrooted. You basically have an information about C and B probably had common ancestor here, and A and B was together here, but you don't know about the root. So it really depends on the interpretation of the tree, really depends on where you place the root. And it is not possible to do that computationally. You have to manually decide where to put the root. And if you put the root, for example, here in A, then it means that from the root, A was separated, just like this one. A is separated, and B, C, D is a clade. So this is one alternative. Or you place the root in, for example, between this part and this part here. Now you have two clades. From the root, you have BC clade and AB clade. So rooting is important. In virus genomes, we are lucky because we have data information and we know all the sequence, and we can use that data information to place the root, because it is probably the most sort of appropriate way of the tree rooting. And now the results are as the following. We used more than 15,000 genomes available in the database. I'm talking about the whole genome sequence of all the viruses. We aligned them all and built tree with them. So this is the 15,000 genomes and their phylogenetic tree. The outer layers show you some clustering. I mean, what I mean is some research groups identified some mutations specific to a group of genomes. For example, in one grouping method, L type, S type, L type is green one, sorry, S type is green one, L type is the pink one. So some claim that L type is more aggressive one than it is refuted by other people and so on. Same thing for spike protein. In this position, some of them have G, some of them have aspartate. Some groups say that one is more aggressive than other, and it is also refuted by others, so on. And barcode, 6 major other clustering methods, but we also applied some clusterings based on the phylogenetic tree only. So we call them phylo-major and phylo-subclusters. In phylo-major clusters, we have 4 major clusters. One is this one, red, this is maroon and blue and purple here, 4 major clusters. And if you look at the subclusters, you will see, I think 17 of them. Why it is important to build tree and see the distribution of the genomes of interest. So we were interested in the genomes isolated in Turkey, and the outermost level shows the genomes isolated in Turkey. So one of the genomes are here, some of the genomes are there, one genome is over here, some of the genomes here, there, and here. So it shows that the samples isolated in Turkey are not clustered on a single branch in the phylogenetic tree. So they are sort of dispersed. And this is an evidence of multiple introduction to Turkey. So the virus, the source of virus was not single, it was multiple, and it is obvious from this tree. Of course, some subclusters are enriched with Turkish samples. Here, this one and this one, for example. But you will see some others as well in the other branches of the tree. And we have observed an interesting case here. You remember S type and L type. So S to L type switch was in China, and then L type was dominant in Europe, specifically in Italy. So remember, our first case was in March 10, but this S type and L type transition was earlier than that, much earlier, because L type was the dominant one in Europe, and Italy had a lot of cases when the first case was explained in Turkey. So we identified one sample as the transitioning version from S type to L type. How do we know that? With 2 things. So first, in the phylogenetic tree, it is placed between S type and L type, which is a sort of clear indicative. And the second reason is that there are 2 mutations defining S type and L type, and only one mutation was in this sample. That's why we can clearly say that it is neither S or L type, it is the transitioning version from S type to L type. And it shows that the first introduction to Turkey did happen before the first explained case, because it was very early, the transition from S type to L type. And we compared the profiles of the countries. Remember our 5 major groups, we had 4 groups, and then we just basically identified the abundance of the groups. So Turkey has majorly the 5 major 4 group, 60%, but this is not the idea. The message of this figure is to show that each country, more or less, have similar, unique distribution of profiles. So why is it important to know about the phylogenetic distributions? Because if you know which are the major virus genomes, then you can build diagnosis tests based on those. So here, Turkey doesn't resemble any of them in this phylogenetic profile of the countries. And then we basically look at the phylo subgroups also. So remember, Turkey was enriched in two phylo subgroups. One is the brownish one, the other one is the blue one. If we just look at the brown one, it is going to be, so y-axis, which is not labeled here, sorry for that, but y-axis is the divergence. How do we measure divergence? From in the tree, we just calculate the distance from the tree to leaf, and this is how we identify, how we calculate the divergence of the leaves. So the brown one seems to be more diverged than its equivalents in other samples collected in other countries. But remember, they are in the same phylo subgroup, so that's why they are comparable. And the blue one is also same. It is statistically significant that it is more diverged. What does it mean? Does it mean that in Turkey there is more divergence? No, it doesn't mean that. It simply means that when you randomly select sequences or genomes to sequence, then it is more likely to choose sequences from these phylo subgroups, and it is more likely to get more divergence from them. So the divergence patterns are not more or less. So averagely we find 2 mutations per month for more or less every genome. So there is no fast evolving or slowly evolving genomes. So this simply means that the major subgroups in Turkey are 2 of them. One is phylo subgroup 4, the other one is phylo subgroup 8. And this shows all 30 samples and samples isolated in Turkey and their mutations. So this is the phylogenetic tree. These are the clusters, and our clusters on right. And this is basically the mutations that we observe, right? So in spike protein, for example, you see this mutation. But remember, this mutation did not independently occur. If you look at the phylogenetic tree, this mutation occurred in the common ancestor of this large clade. So they are not independent mutations. This mutation occurred only once. And if you look at the mutations, you will not see 2 mutations occurred independently of each other. So it also shows that there is no sort of favorable mutation. By just looking at the samples of Turkey, there is no favorable mutations. And these mutations that you observe here are most likely because of the founder effect. So once mutation happened in the common ancestor of these genomes, and it is there in all of them because of its effect called founder effect. And then we identified the transmission routes. I would like to show you 2 videos. This is a general video. So just look at from China, the first case coming to Turkey. So this is around, I would say in January, based on our predictions, in January to February. I mean, not in February, but most likely in January, this transmission occurred. And then we wait some time. We will see other introductions to Turkey. So this is only focused on Turkey, so that you will not see many transmissions here. Only Turkey-associated ones are shown. So you see another one is coming up, and another came from US, and another came from first again US to United Kingdom and Europe, and also from Australia you will see transmission. And in Saudi Arabia, there is one coming to Saudi Arabia, one coming from the Saudi Arabia, and this is how it looks like. So if I just run the same animation here, basically I would like you to see the transmission hub in Turkey, which is going to be Ankara. Let's see how it will look like. So just focus on Ankara here, and then you will see the transmission to the other cities in Turkey. Let's see how it will turn out. Look at Ankara. It's from Ankara there will be transmission to the other cities. So if you just look at here, Ankara behaves as a hub here. We would expect Istanbul to be like that, but remember, this is only 30 sequences that we use. When we have more sequences, we'll have a better idea of these on these transmission routes. This is just a preliminary result. And this is the basically the summary of the transmission routes. I would say that you would see the Ankara being a hub to transmit the virus to Sakarya, Balıkesir, Kastamonu, Nevşehir, Konya. These are the, and also Saudi Arabia. And from Saudi Arabia, there is one associated virus in Afyon identified. And you'll see from North America, there is introduction. From Europe, there is introduction. And from Australia, there is introduction to the country. And then we identified the sequences that should be important for Turkey only. So with 30 sequence, we cannot really say much about it, but we should be careful about some of them, which are only seen in Turkey, right? So in the entire database, there is only one sequence identified. It's another database, and out of 30, 2 of the samples have that mutation. So we should be aware of this mutation in our samples. It might spread more in Turkey, so that's why the mutations are important. So far, the effects of mutations are not shown to increase or reduce the transmissibility of the virus or the fatality rates. There is no report about that. There is no scientific evidence for that. But why mutations are important? If you want to build a test to identify coronavirus, even PCR test or antigen-based test, you should know about the mutations in the genome so that you can develop the appropriate tests for that. Because the antibody that you use in the test may not work with the antigen, with the mutation that we have in Turkey. So we should be careful about that. That's why we want to highlight the mutations that we see an enrichment in Turkey compared to the rest of the world. And finally, I would talk about mutational signatures. So mostly we see mutations from cytosine to uracil. So here it is T, but basically since this is RNA, it is U. T to U mutations are seen mostly. So if you just look at them, this is the signature, right? So why is that? So if you look at the genome of the virus, you will see RNA-dependent RNA polymerase, and there are some correcting mechanism for the mutations. So these correcting mechanisms are nice because virus doesn't mutate that often because of this correction mechanism. However, during replication, I mean the RNA synthesis, mutations can occur. But mutations can also occur in the when replication doesn't occur, right? When replication is not in the progress. So we have a viral defense mechanism, and we have some enzymes. Some of them are called Appleback enzymes. Basically, it converts cytosine into uracil. So this is our viral defense mechanism, and you see C to U mutations as abundant mutations, and we can clearly see that. So why I am showing that? So if this was the mechanism in the previous organism that we got this virus from, then we wouldn't have differential C to U mutations, because most of the C's would have converted to U's already. So why I am showing that is to maybe if we just identify the enzyme that is responsible for that and look for the animals that lack this enzyme, so that we can possibly identify or hypothesize about potential organisms that we got this virus from. And finally, again, final, would virus mutate and disappear? So there are many people talking, and one of the epidemiologists actually used this sentence: considering the current path we are going down, the coronavirus pandemic could still end in 3 ways. These 3 possible scenarios are: 1, people will become immune to the virus, and an effective vaccine will be developed. This is second. Or the virus will mutate and lose its potency with time. So is it possible? So with first two is obvious, right? If we are immune to the virus, it will disappear. Or if we have a vaccine, again, the same thing with the immunity, it will disappear. But with nothing, with no immunity at all, will virus mutate and disappear? This is the question. If you look from the evolutionary perspective, and if you think about this pandemic, there are more than 6 million cases, and each patient has thousands of viral particles. And let's say that's 6 million. Mutation can happen, right? And it can lose its transmissibility. But in evolution, it will lose, I mean, that virus, that particular virus will be eliminated from the nature. How about the rest 5 million something of them, right? So how about them? Will they have the same virus? No. Same mutation? No, because it is disadvantageous for the virus. So the ones will not have, the ones that do not have that particular substitution will continue to transmit. So that's why it is unlikely that virus will naturally have a mutation and disappear, because it will be single virus or maybe tens of viruses. And then it is almost impossible for all viruses to have that disappearing mutation. There should be some evolutionary pressure to have that mutation. And maybe it will find another host, and so that that particular virus will disappear because it found more effective host. So this is a big no. So virus will not mutate and lose its efficiency with time, unless we have immunity. So I would like to acknowledge GIS8. It's a database that we got the sequences from. We appreciate the submitters, and Ministry of Health in Turkey, they are the main submitters of the genome sequences. These are our funders here, TÜBİTAK, the main funder, EMBO, Science Academy of Turkey, and TÜSEP, and Şuli Sabancı University we would like to acknowledge. I would like to thank main people conducting this research. Actually, they are Aylin and Burak are PhD students, Cem and Berkay are master students, Berk, Defne and Zeynep are undergrad students. They have done a fantastic job in this research. And this is before COVID-19, this is after COVID-19. So we hope to get back to our original setting, hopefully sometime in 1 or 2 years, probably. We don't know. I don't want to speculate about that, but this is what we want, hopefully at the end. And in the absence of the students, we, as the sort of people staying on campus, we enjoyed the spring. However, if we had students, I think sharing this beauty would be much nicer. So we look forward to having all of you on campus. So I'm happy to take questions. Thank you very much.
Thank you. And again, today we opened the campus actually for graduate students, so it will be flourishing soon. And there are a couple of questions. First, I would like to unmute Nihal Özdoğan Erol for her question.
Hi, Ogün. How are you? Thank you for the nice. It was I really enjoyed it. I actually asked the question about it's kind of reverse. How do you think in reverse? Would you expect a mutation in human beings actually due to the infection of COVID? How can you speculate about it?
So are you asking, would there be a mutation transmissibility?
Yeah.
Yes, I would say so, because you know, if imagine that this is just regular influenza-like system, right? So if you have a way of coping with the virus, then virus is likely to develop its own mutations and get away from your immune system. So the reverse is most likely will happen. If we have an effective treatment, virus will have a evolutionary pressure on it, and it will mutate. probably the mutated versions will be more advantageous, so that we will see more mutated versions.
So you also you said that you see it attacks the target protein ACE2 on humans. So will that be effect on that protein or something like that?
Yes, it is the connection of virus with the host cell. So yes, most likely spike protein is very important. So, and it really depends on the treatment also. So if you have an effective treatment that blocks the replication of the virus, right? Mm-hmm. So then virus can have additional mutations to sort of eliminate the effect of certain drug. So it also depends on what sort of treatment you have. But naturally, if there's no treatment, spike protein and that connection should be the hotspot for mutations. But if you have an effective treatment targeting other proteins of the proteins, and they are likely to have mutations.
Okay, thank you.
Okay, then there are a couple of questions from the chat from Melik Demirel, and the first question is, how accurate is this map of time of transmission, the error rate?
Right, it is not that accurate for sure. I think it is a large error margin around 20 to 25 days. So within 20 to 25 days, we can predict the time of transmission.
And then the second question is, when was the first time of entry to Turkey based on S and L types? Where are the samples are sequenced in Turkey? Which facility?
Ministry of Health. And it was in Ankara. Mm-hmm. Yeah, this is all we know. And the time is most likely in January or early February.
Okay. And then the final question, how is the transmission through border? Is there any patient data from Iran border, given that second big pandemic in the region was from Iran? Because the routes are shown as the airport hubs. Also, is there a direct flight from US to Ankara? How is the accuracy of transmission determined?
So remember what I said about the missing links. So if you just sample in Ankara, but the original person that the Ankara person got this virus from could be in Istanbul, right? So we are only talking about 30 people here, 30 genomes. Mm-hmm. So in order to establish the missing links, we have to sequence more genomes to be more confident.
And one follow-up question is, can you also speculate about the furin binding site in COVID-19? It has been obtained only in HIV-2.
Sorry, I didn't get the question.
Can you also speculate about the furin binding site in COVID-19? It has been obtained only in HIV-2.
I don't know about them.
Okay. I have one personal question about Ankara. So as far as I know, in the very beginning of the pandemic, all the samples were shipped to Ankara for testing. So could it be a reason? So because Ankara occurs as a hub?
So it shouldn't be, because they should label samples based on where they are isolated from. So based on where the patient was from. So it shouldn't cause bias. But there's a major bias, I would say, that, for example, 60% of the cases should be from Istanbul, but out of 30 genomes, most of them are from Ankara. So that's why it causes us bias, for sure. So unfortunately, we don't have number of samples representing the distribution in Turkey. But I hope that with the more genomes sequenced, we'll have that. Actually, there are some projects going on in Turkey, but unfortunately, the data is not shared so far.
Okay. If there are no any further questions, I would like to thank Ogün personally for this very informative scientific presentation on COVID, because we usually see lots of opinions, but not science. Thank you very much, Ogün.
Thank you for being here.
Thank you, and I'm applauding for everybody.
Thanks a lot.
Thank you very much. We will be hosting a similar seminar in 2 weeks from Professor Berrin Yanıkoğlu, again on a different COVID-related ongoing research. Goodbye.
Bye.